Good afternoon. Thanks, Emma, for the introduction, and thanks for the opportunity to invite me to talk to you today about the work we're doing to try to close the gap on diagnostic odysseys for patients in the Middle East. I'm sure you all know, and thanks, Emma, for giving a nice introduction, that rare diseases are individually rare, but they're collectively common. They can add up to 6% cumulative frequency, prevalence, which means that over 400 million individuals can be affected with a rare disorder at any one point in time globally. They're not easily diagnosable, so patients go through what's known as the diagnostic odyssey, which can be around 7 years, through which they undergo recurrent hospitalizations, unnecessary diagnostic workup. So it's a significant financial and socioeconomic burden. Although we don't have data about the burden of rare diseases in the Middle East, we expect it to be having a similar burden, if not higher. Why? Because in the Middle East, we have a higher prevalence of marriages between relatives, and in some countries, the proportion of first cousin marriages can be up to 50 to 60% of all marriages. The families are relatively large, which increases the risk for having recurrent affected pregnancies. On the other side, there are no genomic diagnostic facilities, so we have a big burden, lack of genomic diagnostics facilities, so you can expect that the diagnostic odysseys can be longer than what have been characterized elsewhere. So recognizing this challenge, our hospital, now a government hospital, was established in 2017 to basically house a multidisciplinary tertiary pediatric center with now over 40 pediatric subspecialties under one roof, which is crucial for rare disorders that often involve multisystem organs. In addition to a genomic diagnostic center in-house that offer clinical genomic testing, mostly based on short-read sequencing, exome sequencing, whole genomes, targeted panels, microarrays, and targeted testing. The center was accredited by the College of American Pathologists in 2019, and we recently published our experience on the first 1,000 patients, who are mostly Arabs from Middle Eastern countries. And although we find that we're able to help a lot of families, the overall diagnostic yield is 30%. It's higher by sequencing, it goes up to 40%, but this still means that there are up to 60% of families that are not finding diagnoses, which is still a major problem. Now, keep in mind here that covering for genetic testing in the Middle East, unfortunately, is not very robust. So if a patient gets a chromosomal microarray covered, and if it's negative, which is 80% of the time, they will have a hard time getting an exome sequencing test, and vice versa. So this brings the motivation to actually establish a comprehensive, accurate single assay that can capture SVs, SNVs, CNVs, and indels. And that's why we collaborated with Oxford Nanopore sequencing to actually have a pilot project where we can sequence, or we are sequencing 50 patients. 10 of them have known pathogenic variants, shown in orange, and then another 40 with negative findings where we expect something missed by short-read sequencing. And like all of you that we've heard over the last day and a half, we're trying to optimize bioinformatics and analysis tools to call, annotate, and filter SNVs, SVs, CNVs, methylation defects. And I think this is still a work in progress, but we are eventually getting to the point where we're able to automate variant calling. And just to give a quick summary of what we are finding now, we are able to detect all types of different variants that have been otherwise detected by different validated clinical assays. That includes trisomies, aneuploidies, UPDs, methylation disorders, SMVs, structural variants. And some of the pipelines are more robust than others, but I think we're getting to the point where eventually we hope to get to be able to automatically call as many of these variants as accurately as possible. I'll run through a few examples. So here is a patient with global developmental delay and an inverted del dupe, tandem del dupe on chromosome 8, as shown by this coverage and breakpoint data from ONT long-read sequencing that's called by QDA and seq. This clearly would have been missed by whole-exome sequencing, but is confirmed by chromosomal microarray, as shown on the left side of this right figure. This is a patient with developmental and speech delay and happy demeanor, expected to have Angelman syndrome, which is a disease where you have a methylation defect on 15q11.2. And as you can see on the top panel, this patient lacks methylation pattern, which usually see as a biparental inheritance of methylation at this locus, as shown in the control lower IGV view. And last case example is this patient who has actually been going through a diagnostic odyssey. The patient had anterior segment dysgenesis, negative testing by single gene sequencing for PAX6, and 3 whole exome sequencing showed an inconclusive result because of a frameshift heterozygous variant that was maternally inherited from the mother in SLC38A8, which is a gene that causes autosomal recessive eye disease, matching the patient's phenotype. But clearly, it's an inconclusive report because it's a recessive disorder and we only have a single heterozygous variant. So doing long-range sequencing, as you can see here, we also identify a 79 kb deletion in this gene, as shown nicely by the breakpoints, decrease in coverage, loss of heterozygosity, developed primers across the breakpoint, and confirmed the presence of a deletion in the patient. And we could do facing. As you can see, the reads are missing in haplotype 2 around this region, unlike haplotype 1, where we also find the maternally inherited frameshift variant. So this confirms compound heterozygosity and also confirms a molecular diagnosis of SLC38A8 in this patient. There are lots of examples. We're wrapping up this work and we hope to be able to publish it very soon. But I know, like many of you, we're getting to the point, and that's our ambition, where we're able to automate and the pipelines as much as we can and replace all different testing modalities in the clinical lab so we can identify all types of variants in one single assay and avoid the sequential testing approaches that we currently have. In the remaining few minutes of my talk, I'll talk to you about another project by Oxford Nanopore sequencing in collaboration with Assuragen, where we are trying to look into SMA as a prototype. Why SMA? Because it's the second most common recessive fatal disorder in several populations. Again, we don't have a lot of data from the Middle East, but from our diagnostic work on patients referred to us from NICU from different countries within the Middle East, we see a high diagnostic yield, around 30%. The importance of this disease is, as you may all know, it's one of the first rare disorders with a gene therapy, which is highly expensive, but yet is life-saving. And with common disorders like this, you always want to think about screening programs, whether premarital or newborn screening. And in fact, it's actually mandated in several states in the US and also in some countries in Europe. And we've started a screening program in the UAE as a prototype for other recessive disorders. And so you always need comprehensive, accurate, and cost-effective ways to test for SMN1 and SMN2. But as you all know, this is a tricky locus. SMN1 has a highly identical paralog, which complicates analysis by short-read sequencing. It also undergoes a lot of frequent unequal crossovers, so you have a lot of variable copy number status for SMN1 and SMN2, which is why the most common pathogenic variant is a deletion. But there are still around 5% pathogenic variants that are SNVs. There's also this configurations where healthy individuals can have 2 SMN1 copies in cis, so there are 2 + 0 as compared to the 1 + 1 configuration. And this has a lot of important consequences for recurrence risk assessments for consanguineous families in the Middle East. So we were lucky and fortunate to be an alpha testing site for a wonderful collaboration between Assuragen and Oxford Nanopore sequencing to develop fast, scalable, and a comprehensive amplicon-based long-read sequencing assay for SMN1 and SMN2. The assay has 2 components, or 2 tubes. The first uses short amplicons, relatively 2 to 3 kb. And in this proof of concept alpha assay, we're only covering exons 3 to 8. It uses, the analysis pipeline uses the coverage data from the amplicon along with the ratio of C to T from this 840 C to T variant that's different between SMN1 and SMN2 to call CNVs. And a second tube also covers those same exons through a longer amplicon, which is around 10 kb. This also uses the coverage information along with paralog-specific variants to infer or resolve copy number status for SMN1 and SMN2. And so we use clinical samples that have been clinically tested in our laboratory. We've tested so far 70 samples, and we find 100% concordance for SMN1 copy number status, and around 97% concordance for SMN2. And I think with additional bioinformatic optimization, we can get this to 100%. But of course, we have also the single nucleotide variants, or the sequence data from those assays. And although we haven't tested a sample with a pathogenic sequence variant, we can look at some of the variants that are, for example, associated with the silent carrier configuration, and we find some of our families in the validation tests that actually have those SNPs and are therefore silent carriers. Here's an example of a family where the father and one of the siblings have those SNPs and are therefore silent carriers, which now explains the inheritance pattern in this pedigree. So have we tested this couple, the mother and the father, using our Droplet Digital PCR clinical assay, which is blind to the 2+2 configuration, we have seen that the mother is a carrier, while the dad is 2-copy healthy, and we would have counseled them that they have a low risk of having a patient affected with SMA. But now knowing that they are 2+0 and 1+0, every pregnancy has 25% chance to be having an affected child, and sadly enough, this family already have 2 affected siblings. I'm skip this for the interest of time, but I hope maybe I talked too quickly. I presented those 2 projects to convince you that long-read sequencing is really has a high potential, great potential of becoming a powerful diagnostic tool for rare disorders in general, and especially for challenging regions like SMA, which is a prototype for other diseases. And I'm sure you've seen Brad Hall's talk from Assuragen yesterday, talking about a multiplex assay with several other complex loci for carrier screening. Finally, we'd like to thank all the patients that we really work hard to kind of try to help them, and they keep us motivated. And I also want to thank the genomics team at Al Jalila Children's who are actually doing the work, and our wonderful collaborators at Assuragen and Oxford Nanopore. Thank you so much.